Hi everyone, welcome to Reason with Science. I'm your host Jitendra. This episode is with Kerstin Göpfrich. She is a leading researcher in the field of synthetic biology with expertise in biophysics, DNA nanotechnology, and microfluidics. She leads the Max Planck Research Group, Biophysical Engineering, at the Max Planck Institute for Medical Research in Heidelberg. Her research centers on utilizing DNA origami, a versatile technique that employs DNA molecules to construct three-dimensional components for synthetic cells. In this conversation, we talk about can we create life in the lab, synthetic cells, advantages of using DNA origami in creating synthetic cells, applications, and the future of artificial cells. Enjoy the conversation, share and subscribe to support the podcast. Thank you for listening. Hi, Kirsten. Welcome to the podcast.
Hello.
So, as a synthetic biologist, so you work a lot with synthetic cells. Let's start with that. Like, how briefly you can describe your work? What do you do?
So I'm a physicist by training, and as a physicist, I learned to I really fell in love with models. So models that describe the world, but in a more simplified manner, in a little bit an abstract manner, right? You may know the model of the atom or whatever we learn in school, right? And I find that really fascinating how you can go about having an extremely complex system, but describing it in an abstract way that simplifies it, and that you can still draw information from it and learn from it. And so when I learned about all of these models, I was always dreaming about having a model of a living cell. Could we build a model of something as complex as a cell? And so first of all, I went into single molecule biophysics. I was building DNA nanopores as sensors at the time, for single molecule biosensing. And I was wondering, well, if I can build nanopores, can I also build other components for synthetic cells, and can I eventually even build an entire fully functional synthetic cell from scratch? And this is kind of what brought me into this field of bottom-up synthetic biology. And really our dream is to build a model of a living cell which will be based on a lipid vesicle, and our ambition would be that it operates based on our own molecular machinery, our own molecular hardware, so to say. Instead of just using the molecular building blocks that nature has provided us with, we would like to make our own as well.
Yeah, that's interesting, and of course we'll talk more about it. But let's kind of also explain maybe. So, what's your model, or how do you envision a cell? Like, what's your analogy once you think of a living cell?
That's a complex question, right? Like somehow if we look under the microscope, we'll always recognize a cell, right? Like this is what we've been trying to do ever since, people have been looking under microscopes, I guess. So of course, if you look under the microscope, you may be looking out for features, like something that moves, something that's divides or so, right? But then again, I don't think that actually collecting these hallmarks or features that we associate with living things and trying to put them back together will actually lead to a living thing, right? So I don't think you can just start engineering hallmarks and then put these hallmarks together, because in a way you run into what I like to call the integration hell, right? So I simply, engineering modules and then trying to make the pieces fit, I think is going to be challenging, simply because life may be more than the pieces of a puzzle. There are emergent properties that we associate with life and so on and so forth. So I feel like this level of complexity will have to actually be an outcome of evolution. So if we engineer living systems, we should more and more start to think about like engineering something as simple as possible, which can self-replicate, self-regenerate, and start to have the capacity to evolve, and then build complexity from there. So if you ask me what is a cell, of course we have the biology definition, right? We have this one example of how a cell looks like in biology 1.0, so the life that we know, but we have no idea what life could be looking like. And if we engineer life, I think this is exactly what we we should start to think abstract. We should start to, go away from life as we know it and just start to think in an abstract way. What are the basic ingredients? What is that we really need? Because in some way, using exclusively the biological hardware is also a constraint, right? Evolution has accumulated quite a lot of garbage over the years, the few years that it's been working on this thing called life, right? So I'd imagine that simpler solutions must exist. And, to give you an example of this, I mean, the whole paradigm of transcription translation, the whole paradigm of, the central dogma of molecular biology that DNA makes RNA and RNA makes proteins means already intrinsically that life is quite complex, because think of all of these components that are needed for in vitro transcription translation or for this whole machinery alone, right? This is quite a lot, right? This means that you need quite large genomes to sustain these kind of activities. But if we think, about engineering our own hardware, we could be as radical, or let's call it crazy as putting information and function into the same molecule, or maybe not the same, but at least avoid part of the complex machinery that nature has evolved. So this is exactly what I mean by engineering our own molecular hardware, coming up with solutions that may be actually quite radically different from, the solutions that life as we know it has. And, the systems we've been playing with have always had to do with DNA nanotechnology or RNA nanotechnology, because DNA and RNA are inherently capable of storing information as well as executing function. In particular, if you think of RNA, with all of these ribozyme activities and so on. And, if you can transcribe an RNA origami, like the group of Eve Andersen has beautifully shown, right? If you can produce RNA origami by just transcription, well, you already avoid the translation machinery, which is intense, which is immensely complex, right? Think of the ribosome alone. So, in this way, we hope to actually look for shortcuts. And it's also an important message to think about, right? Like, I don't think if we think about engineering of life, if we think about this idea of building a cell in the lab, I think there will not be one solution, right? So I completely admire the work of the people who do top-down engineering of cells, and these minimal genomes that have been made. If you could boot them in something like a liposome, you'd also have a transition from matter to life created in the lab. And maybe we are not even, light years away. Maybe this is actually a realistic goal. Similarly, people who encapsulate in vitro transcription translation systems, like, for instance, the group of Kate Alamala, who appeared in one of the previous podcasts, right? I also think this is a completely valid approach to think of making a minimal genome and booting it inside of a liposome in a bottom up manner. So that's extremely beautiful. I just think that there's also a third solution, which is kind of using maybe de novo design building blocks that function a bit different from life as we know it. And I'm still using nature's building blocks in a way. So we are still using DNA, just not only in the context of genetics, we use it also for function in the sense of DNA nanotechnology or RNA nanotechnology. But you could be even more radical, and use completely synthetic components, which is not something we are doing at the moment, but others are. And, in this way, I think we should just not think about just creating life 2.0, but actually 3.0, 4.0, whatever. It will be exciting to see which path is the fastest, which path is has then later on also the most interesting capabilities in terms of open-ended evolution and so on and so forth. So I think there's a lot to explore, and there's no reason whatsoever to be dogmatic about using one approach or the other. The problem itself is just hard enough. So we should just do everything we can in parallel, right? So I think that's the beauty of the field.
Yeah, it is. And one thing that I'm like, it's exciting to know is the fact that you said that we can't really just have modules and put them together and expect that it'll work out. But I think that synthetic biology is about like Lego approach, right? Like we are trying to put Legos together, and we expect that at one time, at one point, like we'll be able to get is some sort of a living system out of it. So
I think maybe that's a mistake. I don't know.
Okay.
No, I mean, I'm married to a systems engineer at Bosch. And you know, if you ask a systems engineer, they will tell you that you can never make a complex system from scratch. And you know, a cell, I would consider it a quite complex system. You actually have to start with and this is what systems engineers tell you, you have to start with a simple working system. And I think in a way the same is true for trying to engineer a cell, right? Like I would imagine that you have to start with something simple, and then evolution has to kick in and make it complex. You cannot expect that if you take complex things and you put them together, that it will work. I think it will just blow up, and you know, nothing will work. So I mean, it's also a message from systems engineering is that basically systems in general work poorly or not at all. So basically, what can go wrong will go wrong according to Murphy's law, right? So I feel like starting with an extremely simple system that maybe just can, you know, self-regenerate, self-replicate in some way to the other, where self-replication is genetically encoded. So you can have evolution act upon it, so to say. This will be the starting point. And this will be the starting point to really build complexity. And this in itself is complex enough, right? So don't get me wrong. So maybe we'll, you know, we have a way to go, a long way to go.
Yeah, and probably to explain the complexity of the system, maybe we can just talk a little bit about the approaches that you mentioned, top down, bottom up. What are these approaches? What's the difference there?
Yeah, so in top down synthetic biology, basically this is the original synthetic biology, I would say, almost, right? Is where people started to actually take genomes of living cells, of bacteria, and made them and looked at what genes are actually essential to maintain life, right? And then basically come up with a set of genes that seems to be essential. So basically, if you knock one more gene out, you know, the system does no longer work. It no longer self-replicates. And I think we are now at 400 something genes, you know, that make up this bacterium from the Craig Venter lab, which can still somewhat self-replicate and self-sustain life. But it's put in a chassis, you know, a chassis that was made by biology. So it wasn't made by us or by self-assembly or anything. It was made by a pre-existing bacterium, so to say. So this is why one could argue that this is not truly a transition from matter to life. Although I think we're getting close, because I mean, after all, this bacterium was stripped off its genome, so and a new genome, a synthetically made genome, was put in. So I think this is actually quite cool. And in this top-down synthetic biology approach, you know, the dream is really that we can start to engineer in other genes that are you know, doing useful things for us, producing chemicals, producing medicines, or whatever. So I think this is a really fruitful thing to do. And in the bottom-up approach, you know, we start with yet simpler systems. So we really say, okay, we start only with matter. All of the components are isolated, are purified, and we really start to try and put them together to build a cell from the bottom up. So really radical, really, you know, in a way, yeah, making a cell from non-living building blocks. That's kind of the vision of the bottom-up approach. And the beauty is that you understand everything, you hopefully understand everything you put in, right? So each component, you know, you need it for this, you need it for that. It's in precise quantities, you know the exact composition of your cell. And in a way, this is what we want, right? Full understanding of the system and, you know, the minimal complexity possible to have a self-replicating cell, so to say, and then, you know, let evolution kick in. And I think if we see signs of open-ended evolution of greater complexity in these kind of systems, this would really be a dream. So I feel like we are now at the point, actually, where the field bottom-up synthetic biology has been extremely focused on rational design. It's beautiful, right? I mean, it's the same for the field of DNA nanotechnology. When we realize that we can build smiley faces and other things out of DNA, this is just, wow, mind-blowing, right? So you because you can, you start to rationally engineer things, right? And this is what happened in the field of DNA nanotechnology. This is what happened in the field of bottom-up synthetic biology. We are rationally engineering our systems, but eventually we have to realize that you can only rationally engineer as long as structure-function relationships are known, right? So you need to know what you engineer and how you can approach this problem. And I feel like we may now be reaching the horizon of known structure-function relationships, especially when I engineer components for synthetic cells with DNA or RNA nanotechnology, and I want to engineer complex functions, then I don't always know how to do it. And I think there we can really look at a different field, namely directed evolution, right? Because in a way, directed evolution is the opposite of the design of the rational engineering paradigm, which basically let randomness come into our systems. And this is really powerful. It's in a way exploiting what has been so powerful in natural evolution, right? So, and I feel, starting this now, our approach also to say, okay, we have to start with something that's rationally engineered, that's working somehow, but it's actually quite shitty, and then let evolution brush it up, right? I think that's going to be an interesting approach, and I'm curious to see how it goes.
Exactly. I mean, so you mentioned about the cell which was made at Craig Venter Institute, and so the minimalistic bacterial cell has about 473 genes, and it already shows quite stripped off functions in a way. Mm-hmm. And so that already shows that means a cell is not only the components it has, it's also this evolution, the history which it has gone through, right? I mean, simply just because it is embedded in the system, we can't really go like to a minimal extent as we would like to go. But on the other hand, once we use this bottom-up approach, we can also control this evolution period. I mean, we would know how much or what are the paths taken to reach a certain function, right?
So the way I think about bottom-up synthetic biology is a little bit to try and replicate, but not replicate. Let me explain what happened at the origins of life, right? The origins of life, at some point we must have had very, very simple self-replicators that maybe got compartmentalized, that maybe started to genetically encode the division, the replication of the compartment or something like that. I have no clue. Nobody knows what went on at the origins of life, right? However, what is clear is that complex life evolved from simple constituents. And I don't want to replicate exactly what happens at the origins of life or find out what exactly happens at the origins of life. But I think if we build life 2.0, if we build our own versions of synthetic cells, we also somehow have to start with something extremely simple. And then we get the rest for free from evolution, so to say. So this is my strong belief that, maybe by starting with the already evolved transcription translation machinery, you're actually setting yourself quite a hard task, right? But if you kind of start with something much simpler and then let evolution kick in, then maybe you're actually quicker at ending up at something which deserves the name alive. Maybe you don't. I don't know. It's just a different approach. So the way I think about engineering cells is more like starting from something extremely complex. And the reason being exactly, what I told you also about systems engineering and so on, that complex systems just are doomed to fail. So start with something simple and build complexity in an evolutionary process is my credo, my approach there.
Yeah, and maybe here we can just like extend and we can talk about your approach where you use DNA nanotechnology. So interestingly, all the life forms, they use DNA as an information, right? This is what we know, at least so far. But you have decided to use DNA not only as an information storage machine, but also for the building blocks of a cell, right? So, what is your approach? How you are exploring DNA nanotechnology?
Mm-hmm. So, I mean, first of all, I should make it very clear. I think there's no reason whatsoever to be dogmatic about your choice of hardware, so to say. So, DNA nanotechnology seem to be useful for us for the moment. It's also, what we can do well. But I think it's also part of the fun, and it's really necessary to keep scanning the horizon for new tools as they come up, right? Like, there's no reason not to use 3D printers to make synthetic cells. We've played around with those, right? There's no reason not to use, protein engineering, RNA nanotechnology, whatever new tools may pop up. Directed evolution is an important one, microfluidics maybe, So whatever new tools pop up, I think let's just be open, because our task is complex enough. So make our lives as easy as possible. For me, DNA nanotechnology is quite useful because it, first of all, as I said, like DNA is capable of encoding information, but it's also capable of carrying out certain functions. And we were able to build functional architectures like DNA nanopores, like mimics of DNA cytoskeletons, like DNA structures which cause fusion of vesicles, the structures which can shape vesicles, or can alter the phenotype of our synthetic cells, so to say. So this is why we learn quite a lot from interfacing DNA nanostructures, functional DNA nanostructures, with membranes. The other reason to do this is simply because while we go towards our dream of building a synthetic cell, we also build tools that are actually useful for cell biology, right? So if you can remodel a vesicle with a DNA nanostructure, you can likely also remodel a cell with a DNA nanostructure. And then you can start to ask questions, how do certain membrane properties like membrane curvature influence processes like endocytosis or...
So I think DNA nanotechnology is certainly one tool out of many, right? It's part of a larger toolbox. And the reason I find it useful is because you can build components for synthetic cells, but you can also directly use these components inside living cells as biophysical tools, right? So think about, for instance, questions like, how does cell-cell adhesion impact collective cell migration? We can use the same linkers that we use to link our synthetic cells or lipid vesicles. We can use them to link cells and, engineer their binding strength and so on, which is something that would be actually quite complex to do with protein engineering. So all the tools that we build towards our big vision of building a cell, they're directly applicable as biophysical tools in biology. So this, for me, is another reason to keep developing these kind of tools. And yeah, as I said, DNA can encode information, but it can also be functional. Even more so, I would say this is true for RNA, because right now I have to say one big flaw of our systems is that quite often we need chemical functionalization for function, right? So for instance, for membrane spanning DNA pores, you need to attach cholesterol tags in order to make them self-assemble into lipid membranes. Or if you build a cytoskeleton, and as we did, and you want to assemble it in a reversible way using DNA, we had to put azobenzene as a photoresponsive molecule into the DNA duplex. And this is, of course, chemically challenging. It's a synthesis that can be done, but it cannot be done inside a living cell or inside a synthetic cell, and probably never will be, So this means that we would, of course, love to genetically encode our structures, also because then we can start to use directed evolution to make them better, or use directed evolution of vesicles to improve the function. But, if you require crazy thermal annealing, or if you require purification, or if you require, in particular, chemical functionalization for function, then this is not really compatible with the chemistries or with the environment that you have inside of a synthetic cell. And so this is why I think RNA origami, RNA nanotechnology is even better. So what we are now trying to do is kind of transfer the knowledge that we gained already working with DNA also to RNA structures.
Yeah, this is exciting. And so what do you mean by DNA or RNA origami?
So DNA, let's start with DNA origami. So DNA origami, or the field, DNA origami is a subset of the field of DNA nanotechnology. DNA nanotechnology is really about repurposing DNA as a construction material. So taking it out of its context from genetics and using it purely for architecture. And this is based on the famous realization by Nat Seaman, who realized in the 80s actually that you can use nucleic acids to build stable nucleic acid junctions. So anecdotally, this was when he walked past one of the Escher's paintings that showed these kind of fish in a crystal-like arrangement. He walked past this painting and thought, what if we replace the fish with DNA double helices? And, you know, this is what he did. He envisioned that one would be able to use this for protein crystallography. So he was actually a structural biologist who was interested in crystallography of proteins. And as we know, some proteins are challenging to crystallize. He thought, okay, what if we actually place them in a crystal lattice built from DNA? And this is when the field of DNA nanotechnology was born. And sadly, Nat Seaman passed away some time ago. And, you know, we really have to give him full thanks for establishing such a collaborative, such a, you know, thriving field in science that, you know, is all based on these really brilliant ideas, I think. So that's the field of DNA nanotechnology, is about constructing with DNA. And then I would say it really took off or kicked off when Paul Rothman developed the DNA origami technique, which is basically a specific version of DNA nanotechnology where you take a long single strand of DNA. It's actually taken from a virus, a viral genome, or the genome of a bacteriophage. The M13 phage quite often. It's a long single strand of DNA that you can isolate. It's got 7,000 to 8,000 bases. And then basically you take this genome and you synthesize short synthetic oligonucleotides of maybe 50 bases or so that can bind the long single strand of DNA in specific positions. So imagine you've got a long single strand of DNA, you sequence it, and one end has a certain sequence, the other end has another sequence. So now you can envision designing a short piece of DNA which is complementary at one end and complementary at the other end. And then you pull your long single strand together in a loop. And now if you take more of these short pieces, you can start to fold the long piece into more complex shapes. And like the famous smiley or other DNA origami shapes that have been made since. So DNA origami, really with 200 short strands in this viral genome, you can build finite sized objects on the nanoscale. And the beauty is that each and every of these 200 oligonucleotides has a unique sequence and a unique position within the DNA origami. And this means you can use DNA origami also as a molecular breadboard to position other components, you know, be it fluorophores. So you can use this for standards for super resolution microscopy, for instance, or be it antigens to position antigens at a given spacing or valency to improve immune response or to probe immune response. So there is really a vast variety of applications that the DNA origami technique has by now. And synthetic biology is certainly one of them, an interesting one to try and, you know, engineer molecular hardware for synthetic cells. Yeah. And RNA, so that's DNA origami, right? RNA origami, you could naively think, well, it should just work the same way, right? Also, RNA can base pair, and why not just take a RNA strand and fold it like you would fold DNA based on canonical basis, so to say, based on, you know, A binds C, A binds T and C binds G, right? But actually RNA is much more flexible in terms of the structural motifs that you have at hand. RNA can form kissing loops, RNA can form dovetails. So actually the design paradigms with RNA are actually quite different from DNA. This is what we are learning right now. And the beauty of RNA is you can design the structure such that they fold a bit more like proteins do, right? With DNA origami, what we do is we design the sequences, we put them all in a pot, we heat them to 80 degrees, we cool them down slowly so that they can basically thermally anneal to find their energy minimum. With RNA, the beauty is that you can fold an RNA origami from a single strand of RNA. So basically more like a protein would fold from a single chain of amino acids. You can also fold an RNA origami from a single chain of nucleotides, so to say, making use of this also non-canonical structures like dovetails or kissing loops. And this means that RNA origami can really fold in a co-transcriptional manner, just like proteins fold co-translationally. And this is really beautiful work by the group of Elbe Anderson and others who worked on this co-transcriptional folding. And I think this will open up a myriad of new opportunities to really design and genetically encode functional structures also for synthetic cells.
Yeah,
and it is really new. I have no idea.
Yes.
We're just starting to explore it. So, don't quote me on this, but we'll see in a few years, I guess.
Yeah, I mean that's fine, and I mean that's the exciting part, because I'm totally with you in terms of when it comes to like what we can engineer to like and what we can incorporate in synthetic cells, for example, because definitely nature has explored certain chemistry, but that doesn't mean that's the only chemical space that we need to stick to. I mean, the kind of systems that Stephen Benner is exploring or Lee Cronin is exploring, that they want to go really at the basic level and just try out with any possible chemistry and see if we can kind of get what you said, life 3.0 or 6.0 or whatever.
So, And I don't know, like I think Kate was saying that, first we have to work with what we have, which is in a way, it's a fair assumption to say, okay, like we kickstart our systems by using what is there already. We make our lives easier. We make a life 2.0 that works kind of like life 1.0 does, and then we go from there and get crazy. And the other way is to say, no, actually, we may approach it from the other side and think, okay, how simple of a solution can we imagine? And this may not be the life that the solution that life came up with, right? Like evolution is not designed to make something simple. I mean, the life that we have is extremely robust. One really has to say this, right? It's solutions exist for the most diverse environments, right? So maybe and this is also, by the way, an interesting lesson from systems engineering, that every system is part of a larger system. So of course we cannot think of life without thinking of its environment. Every living system has to function in a given environment. So I mean, we need essential amino acids. We cannot live without food, and we cannot produce everything with our own bodies, right? Other types of cells are maybe a bit simpler. They need maybe even their lipids and cannot produce all of their lipids, like mycoplasma, right? You need to feed them certain lipids so they kind of act as parasites, right? Parasites need their hosts to work, viruses even more so. Like they need a whole host cell and basically capitalize on their machinery. So I would say, maybe our idea in synthetic biology of making everything autonomous is also a little bit too ambitious, at least for the beginning. So I would say in the beginning, our synthetic cells will really be as simple as possible and function only in an extremely resource-rich environment where we resupply all the reagents that are needed at the right time, so to say. So we are kind of helping to kickstart this process of evolution. But then once we are there, we hope that evolution can take care of actually maybe getting rid of some of the components and, increase the degree of liveliness or decrease the degree of complexity of the environment, right? So it's also an interesting concept, I think, which is getting more and more established in the field, that we shouldn't even talk about, is it alive or not? We should talk about the degree of liveliness. And somehow, to me at least, and I don't know if this is entirely true, but it seems to be that degree of liveliness is correlated with complexity of the environment, negatively correlated, right? Inversely correlated. So the more complex the environment you need to sustain life, the less alive something is, the more. So a virus, for instance, I would put very low on the liveliness scale, right? Because it needs an extremely complex environment to self-replicate. Whereas something like us, we are kind of capable of living in, we need less components, so to say, to maintain processes. Or let's not even talk about us. Maybe it's taking it a step too far. Let's talk about a cell, right? It requires a bit of a less complex environment, but it still requires an environment. There is no, as I said, there is no system without a larger system around it, right? Life does never happen in a vacuum. There's always an environment. And this is an important message, I think, when we talk about what is an autonomous system. So at the beginning, our synthetic cells will not be entirely autonomous. If that would be our ambition, maybe we should stop.
Well, I had a question about that. I mean, what does self-sustaining means? Because, if someone asks us, this is what we can reply, that we want to build a self-sustaining system, but,
self-sustaining chemical system capable of Darwinian evolution.
Exactly.
It's like a basic definition or one possible definition of life. Yeah. I mean, self-sustained to me means that at least one component is self-sustained. So that's the simplest, that's the simplest way of thinking of it. So one component has to be converted into something useful and waste. Ideally more than one component. Most likely more than one component will be needed to self-replicate the entire thing that you need. But I would say, start as simple as possible, and if somebody makes a cell which operates based on, input of everything more or less, as long as you still, sustain self-replication in that way, maintain an out of equilibrium state over a prolonged period of time and create a system which is capable of evolution. I mean, why should we point at that person and say that this is cheating, right? I think there's no reason to. Because as I say, of course, this is not the end goal. I'm not saying that we should stop there. Not at all. In the end, we want exactly that. We want a system which can do much more complex things, self-regenerate many, many more components, do exciting things, even surprise us maybe, right? This would be the coolest thing if my system surprises me and comes up with entirely new solutions to problems. But this is step 2. Or maybe 3 or 4, whatever, but maybe not step 1.
I mean, I draw a lot of parallels to what is happening in the artificial intelligence field, for example, right? I mean, there we can say that the whatever systems that we have, it's not like human intelligence, etc. But still, we are continuing. People are going ahead with the research and the kind of tools that we have developed. It's already amazing, right? And that and those tools are basically silicon based. They are not using neurons or biology that we have now. I mean, we are going more into we are trying to find this quantum computers, etc., which will make it more complex. So at the end, I think that this is also, or this should be also an inspiration for synthetic biologists, that we need to kind of just continue with the different ideas to see what we can end up with, right?
Absolutely, and I like this AI analogy, right? Like in a way, a synthetic cell is like a synthetic cell that's evolving is a physical manifesto, something like AI. I mean, I would say, and I mean, the question is, to what extent also should we be using these approaches, right? I would say, okay, we still want to build a chemical system, so life that's only happening in the computer, I would say, I mean, is also a very interesting, line of research, but maybe very different from what our community is doing. However, of course, when I say we use directed evolution to let our systems be brushed up or perfected or improved, of course, we can also use AI to help us with this, right? So So I think there's certainly crossovers, and doing simulations, and this is also again a paradigm that comes from systems engineering. Use modeling, use simulations to predict as much, to study your systems in as much detail as possible. So certainly modeling approaches, simulation approaches, also using machine learning and AI are very, very valuable tools for the field, and we should be using them. So I listed some tools before. I didn't say AI, but I clearly should have. Yeah. It's an extremely useful tool for us. And this doesn't mean that we should go completely in silico. I mean, I still want to have a cell which I can look at under the microscope, so some kind of biological system capable of self-replication and evolution. And this is also, I think, this is not also, a crazy ambition or so. I think it would also be cool to use biology for construction, right? So can we engineer synthetic cells which will grow things, in our everyday lives, which will grow, the way we use solid state materials for construction right now? Can we use cells to build things for us, to do things for us, right? So in a way, I think this will also be a revolution in the end for manufacturing, for produce, in the very long run. For now, we should think about, second use of the tools. So as I say, of course, you're constantly asked by funders, or we are constantly asked by funders to justify ourselves, right? Why are we building a synthetic cell? And I have to say, my honest answer is because I'm curious. I want to know what it's like. Can it be different? Is it possible to have a transition from matter to life in the lab? This is my honest motivation for doing this. And there might be something that comes out at the end that is extremely useful. That's a complete paradigm shift in the way we construct things. Yes. But right now, all I can say is that the tools that we are developing towards this are directly usable as tools in biophysics to manipulate cells as they are, to understand more about biology, or if it is, so for instance, when we were working on synthetic cell division, we were looking at mechanisms, at physical mechanisms to make cells divide. We eventually realized that we had actually built a powerful osmolarity sensor which works in solution, right? So current osmometers require freezing of the sample, which is obviously not compatible with live cell microscopy. So we patented an osmolarity sensor, which is based on lipid vesicles, which, can work under a microscope. So maybe it's an interesting parameter for cell biologists to look at. If we had thought about building an osmolarity sensor, probably we wouldn't have at all, but maybe at best I could imagine that we would have improved like these pre-spawn osmometers that already exist, right? But coming up with something that can measure something in an entirely different context, sometimes you just need to do this blue skies research, and you stumble across solutions that you didn't expect. And this is really my message, or the only thing I can say to support blue skies research is that I think I fundamentally believe that it will pay off in the end. It will come up with solutions that we don't expect. It's like, Henry Ford said, if I would have asked people what they want, they would have said faster horses, but then he came up with the car, obviously. So, sometimes real innovations cannot be planned for. They are just you do something else and you come up with something else, right? Like particle physicists did, at CERN, and came up with the internet, right? So sometimes these things come out of the blue, and I feel like it's completely justifiable and valid to do these kind of blue skies research towards building a cell, because A, the end goal may be revolutionary, but B, the tools that we use are directly usable, or the tools that we develop are directly usable, and we just have to keep our eyes open, right? We just have to make use of these opportunities.
Yeah, engineering is awesome in that sense, right? I mean, I was talking to Robert Aumann, who got a Nobel Prize in 2005 in economics. And so he was working, he said that he was working on something like knot theory in mathematics in like 1960s or 70s. something like that.
Yeah.
So he's like, this was a project for my PhD, and it was, I got to know, and it was pointless. Like, who cares about knots and how many knots are there, or what turns you need to, you should use, etc. And he's like, but this was my PhD. I did it, and I could explain about like what turns and exactly what do you need.
Yeah.
Okay. And then this thing, he's like, after 40 years or something, he was sitting at home and his grandson called him. And he's like, I want to ask you about knot theory. And he's like, you are studying medicine. So why do you need knot theory, which is like used in mathematics? So he's like, the knots, like DNA sometimes form knots. And they are teaching us in the class right now. So he's like, maybe I can ask my grandfather about knot theory. So something really basic, but you never know like where the application of basic science may emerge, right?
Yeah, absolutely. I mean, that's a fascinating story. Yeah.
Yeah. So since you mentioned about different technologies, the other technology that you are using is this laser printing. That's also quite interesting. So tell us a little bit more about this.
I can tell you the anecdotal origins of this research. So I first arrived in Heidelberg as a Max Planck research group leader in 2019. And when I arrived, there was a cluster of excellence at Heidelberg University and Karlsruhe and the KIT on 3D meta made to order. And this cluster was on laser printing, developing laser printing technologies. So when I arrived, they asked me, do you want to be part of the excellence cluster? And of course, as a young new PI, this was extremely attractive, and I wanted to, get to know the community and so on. So I said, yes, but you have to do something with 3D laser printing, they told me. So come up, think about it. And I thought about it. I thought, okay, I've never had anything to do with 3D laser printing. So what could we be doing with it? And I thought, well, somehow actually in bottom-up synthetic biology, we have a little bit this problem, like you may know the bottle in a ship, right? Where you put a bottle in a ship, but then it becomes very, very hard to assemble that ship inside the bottle, right? In synthetic biology, we have a bit the same problem, right? We have some sort of a compartment. We can put molecules inside, but once these molecules are inside the enclosed compartment, it's very difficult to get in and manipulate them, position them in space and time, right? And then I thought, okay, we have 3D laser printing. So with 3D laser printing, we should be able to access the interior of the compartment and actually print structures on the inside of this compartment. Turns out nobody had done this so far. And when I presented it, there was also a little bit, let's say carefully, a little bit of doubt that this would work because of various reasons. But a master student, Tobias Aveli, he picked it up and made it work in no time, actually, and then started to print structures on the inside of lipid vesicles using 2-photon 3D laser printing. And, this is super exciting because we were able to build up structures on the inside of lipid vesicles that was something that we couldn't really do before. And some of the structures we were also thinking about functions. So we were, for instance, printing a nanopore across the membrane of a lipid vesicle, right? And then we started from there to develop actually resists that are more interesting for us in the context of synthetic biology. So we started to develop DNA-based photoresists by introducing photocleavable groups into DNA. And then we were also able to print DNA on the inside of a lipid vesicle. That was done by Tobias Walter, also a master student at that time in the group. They are both PhD students now. So that's that was a fun tool to play with. I also think that, we won't be 3D printing the final synthetic cell, but again, it is a tool that's extremely useful for us. So we are now thinking into directions, of course, using this for cell biology as an interesting tool, but also we are thinking now about, the laser printing as a method for selection and sorting, actually. So other types of applications, but again, something that emerged from a complete blue skies idea, and that we hope will find its applications in various fields.
Yeah, it's fascinating, and maybe, who knows, in one day, if we can really print synthetic cells, that'll be amazing.
Yeah,
Okay.
I mean, printing something that looks like a cell will not be so challenging, but building something that functions like a cell will be, right? Like a static printed structure is not so helpful for a living organism, but it could definitely be a starting point, right?
Yeah. Has someone tried to print, for example, just the vesicles? So looking at the membrane organization, for example, can it assemble in the same way as it would in the solution, for example?
So if you ask whether we've tried to print compartments, like to print basically a hollow sphere, right?
Yeah.
Then the answer is definitely yes. Mm-hmm. So printing a compartment in itself is possible. And this compartment is probably tunable in terms of its porosity when you tune the resist composition and so on. That could be a strategy to actually print a compartment. If you ask whether, we can print a lipid vesicle, I mean, then why not just use self-assembly, right? Lipid vesicles or lipids self-assemble into compartments themselves, right? So in a way, maybe, it's even overkill to think about think about printing this, and I wouldn't also see completely how that would work. So.
Interesting. So what are the components that you can build using DNA origami, for example, or RNA origami?
I mean, so let's look at the components that have been built from DNA origami. So definitely transmembrane pores, the group of Fritz Simmel, group of Ulrich Kaiser, where I was a PhD student, and others have shown that you can basically functionalize pores that are made from DNA origami with cholesterol tags or other hydrophobic moieties, and then they will self-assemble into lipid membranes, and you can tune the pore diameter, you can tune things like, the conductance of the pore, and some of those pores have been shown to transport single-stranded DNA or proteins or so. So it's a customizable pore, which I think is quite cool, right? Other components that have been built from DNA origami or DNA nanotechnology for synthetic cells are these DNA filaments by us, by the group of Elisa Franco, for instance, and others that made these DNA filaments that have different types of functionalities or DNA nanotubes that mimic functionalities of cytoskeleton. So like reversible assembly, or they have been modified with cholesterol tags to deform the compartment. We have, for instance, included azobenzenes, the Kevin Jahnke in our group, to make them assemble in a reversible manner, or we've modified them so that they can transport cargo along the filament. So these are possibilities that we can engineer with DNA nanotubes, and then others have built mimics of snare proteins to make membranes fuse. Others have built scramblases, so basically structures that self-assemble into membranes and transport lipids from one leaflet to the other. Then there is a variety of structures, for instance, also from a group of Petra Schwille, and also we've used some of these structures where we can deform lipid membranes. So we can alter the shape of a lipid vesicle. We think of our synthetic cells are mostly spherical, which is kind of boring if we think of the phenotypic diversity of cells, right? So we started to deform lipid vesicles with DNA origami or DNA nanostructures. So there are really a myriad of opportunities. And I mean, if you think about it, what are really the ingredients that we need for a very basic cell, then, we are in a way already much further, right? All we need is, in a way, growth and division, and this all over, right? Self-regeneration. So in a way, what we need to do is basically develop DNA nanostructures which can change the curvature of a lipid vesicle such that it can divide. And then other DNA nanostructures maybe which allow the vesicle to regrow. Or maybe we don't even need a vesicle. There are beautiful works by, for instance, the group of Andreas Walter, who've looked at, making entirely DNA-based compartments. So, DNA droplets is also something we've been working on to, for instance, make a DNA droplet as kind of as a mimic, as a nucleus, which can then self-divide by photocleavage, for instance. This is the work of my PhD student Mai Chan, who was looking into, whether we can divide DNA droplets inside of a lipid vesicle and use this potentially as a kind of a, information storage that can be divided together with the compartment. So you see, it doesn't always have to be a lipid vesicle. It can be an entirely DNA-based compartment which could self-replicate. So I think, as I say, I think we can be open in terms of the hardware that we use, as long as we get towards our end goal. We like to work with lipid vesicles, nevertheless, because I think lipids are quite fascinating.
Yeah, and the fact that it's just simply because of the physics assembles in the vesicles, right?
Yeah, and also simply because of the physics, you can actually get structures which are very closely, related to division, right? You can basically, as you move on this shape diagram of spontaneous curvature and reduce volume, so basically osmotic deflation, you can get a variety of lipid shapes just based on lipids alone. And I think, if you help a little bit with DNA, RNA, nanotechnology to get full division and to get regrowth, I think we can really get towards something that, or I hope we can get towards something that deserves the name synthetic cell in a way.
Yeah, but then how far you can go with DNA, RNA, nanotechnology? Like, can you build everything out of it? What's your take on it?
So, I mean, this is a complex question. What is everything? I mean, I could definitely see that you could build something that deserves the name cell, based on, lipids and RNA, DNA alone, right? That, and this is an interesting question, right? Does life require proteins? Yes or no, right? And the systems that we are building are at the moment using proteins, to for instance, produce RNA from a DNA template, right? T7. But, if you look at templated ligation or so, or other mechanisms, maybe this is not needed. It's just that you make your systems extremely slow. And, you could imagine that an environment, even a probiotic environment, proteins were just floating around potentially, right? So I don't know. It's a bit of a cheat to use proteins, but, I could imagine a life without proteins, let's say. It would probably be much slower. So in direct competition compared to a life with proteins, it may, be losing out, right? But without competition, I could imagine that systems like this, that you could build a self-replicating evolving entity from, without protein in a way. As you increase complexity, maybe proteins come out, or are needed, right?
Yeah. So you've been mentioning that we can use DNA, RNA, origami, or nanotechnology to build hardware. But what is, for example, software for you? Is there a software in the cell?
Well, I mean, I don't know. You could argue the software is the genetic code that's, I mean, but yeah, in a way, okay. In a way, what you need for the cell is some form of information encoding, and then molecular hardware, which translates information to function. And then evolution can act upon the function, and then the whole system is in close, is communicating with its environment, and the environment is what puts the selection pressures on the system, right? So we need some kind of molecular hardware which translates information to function. Whatever this information may be, whether that's DNA or something completely different. By the way, I would love to use synthetic polymers or so, right? It's just, you have to be extremely an extremely good chemist or extremely rich, So I think, just on the scales of typical research budgets, it's just, DNA is quite a useful sequence encoding polymer to use. That's the reason for us to use it also, because it's so well understood. But of course, there's no reason not to think of something completely synthetic.
Yeah, I mean, since I work with proteins, so I've been asking this question. For example, when it comes to origin of life question, generally especially once we think of hydrothermal vents, right? The idea is that the simple or prebiotic chemistry was driven using just the metal ions, the metal surfaces at hydrothermal vents. So I've been asking this question, then why do we need proteins? Why, like, if the simple metal surfaces can catalyze the reactions, then why do we need enzymes to do that, right?
In a way, I think the answer is because they are faster, right? So as soon as you have enzymes, they will take over. And then there's no way going back, right? Maybe. Yeah. But I think we have no way of knowing what exactly happened, at the origins of life. We can only think, okay, what can we do to make life 2.0, life 2.0, 3.0, 4.0, whatever. So yeah, that's a really fascinating goal, I think, to see, can we achieve a transition from matter to life in the lab? And I think this is really something I wish I witnessed on the scale of my scientific career, right? That would be really a dream, to be at this forefront of knowledge when it actually happens, and when we are successful as a community to make this happen. And the question is, will it ever? And I tend to ask this question at the beginning of each and every talk I give, whether that's to high school students or to my colleagues in the field. I ask always, if you want to build a cell, do you think it's even possible? And it's quite interesting, because there seems to be this, almost like constant percentage of 90% of people who think, yes, it's possible. And there are about 10% of people who are a little bit in doubt. And that's true whether you ask high school students or the experts in the field, right? It's really interesting. So most people think it is actually possible. And this makes me, of course, very happy and hopeful. I'm one of them. 10% of people say, no, you'll never succeed. Which of course would be a pity. Then I dedicated my scientific career to the wrong thing, but I think it will be possible. And I think there's been an interesting argument made that, in organic chemistry, people who are at the same point at some point, right? Like they said, okay, an organic molecule like urea can only be produced by living things, right? And then eventually urea was synthesized in the lab, and this kickstarted the field of organic synthetic chemistry, right? And I hope in some way that we are at the same point now in synthetic biology, that, proof of principle is needed to really kickstart an entirely new field of science, and a very exciting one, I feel.
No, in my conversation with Nick Lane, he mentioned something interesting. He's like, as a scientist, you have to doubt yourself every day, but then still you need to push yourself to believe in what you're doing. So it's also like that, right?
Yeah, you have to somehow believe in the big picture, or just be so fascinated by the big picture that, even on a daily basis, when you doubt things and things go wrong, right? This is completely normal. Things go wrong in the lab, that your love or your vision of the big picture keeps you going, right? In a way that, even if you doubt yourself because some things don't work, you still believe in the bigger picture. And I think this is actually one of the beauties of being a PI, that of course there are several projects going on in the lab in parallel, and even if something is a little bit stuck, you have maybe other things that go really well, and then you can try and support the people who are at the moment struggling more towards so then you've got 10 hands all of a sudden. Things are moving faster than I would ever have imagined. It's really nice to see.
Yeah. One thing that we didn't talk about so far is the powerhouse, or how the life is powered, basically metabolism. So What's your take on that? Is there any progress in using DNA nanotechnology to build metabolic systems, for example?
Yeah, so I mean, so that's of course entirely true. Life is an out of equilibrium process, right? Like we have to maintain out of equilibrium conditions in our systems. And again, I mean, in the end, what does nature do? Nature is converting, for instance, a proton gradient into ATP synthesis, right? So in a way, proton gradients stand at the beginning, and so gradients you could imagine by providing them by external means at the very beginning, right? So you could basically use microfluidic systems to maintain gradients, be it temperature gradients, be it proton gradients, or whatever you may want to use. So in a way, you can artificially fuel your systems in that way. If you think about building an ATP synthase from DNA origami, then I have to disappoint you. There is no version of ATP synthase that makes ATP that we've built entirely synthetically yet. However, there are DNA origami-based motors that have been developed by the Simmel group and also by the Dietz group, where DNA origamis can spin around, for instance. So there's definitely work going on in this direction. At the same time, I have to say, I would again say that in a way we are at the point of unknown structure-function relationships, right? If we say, okay, can we construct a protein pump from RNA origami, right? Maybe we can, I just don't know how. But I think there may be ways of moving also that field forward. But again, as a start, I would say let's just fuel our systems externally, or reconstitute. This is also what people do in the field, right? They reconstitute proton pumps and ATP synthase and lipid vesicles to shine light on the systems and produce ATP like that, right? And that's extremely beautiful. And yeah, the question is just, can you self-regenerate these, and then it becomes challenging again? Or do you need to supply them from the outside at the beginning, which I think is a fair solution to start with. Not a good end goal. We are ambitious, of course.
Yeah, and you've been also working with something like synthetic eukaryotic system. So what's the difference? What is the complexity there and how you are trying to build it?
I mean, as I say, first of all, we want to build a cell, and whether that's prokaryotic or eukaryotic or nothing or everything, I don't care, right? So we are not even at the point to give it a name like that. Of course, you can try and mimic features or hallmarks of prokaryotic or eukaryotic cells in a bottom-up synthetic biology approach. So one hallmark of a eukaryotic cell would be, for instance, internal compartmentalization, right? So we could put internal compartments on the inside of our lipid vesicle, like we did, for instance, with these DNA droplets that we form on the inside of a lipid vesicle. It's kind of an internal compartment that mimics the cell nucleus in a way that's, you would only find in a eukaryotic cell. Or you could think about, adding a cell wall from the outside with DNA nanotechnology, like kind of make a shell around the vesicle. It's also something that has been done that mimics maybe the cell wall of a prokaryotic cell, of a bacterium, right? Or you could make a cage which gives it this characteristic bacterium shape also, right? Again, this is engineering hallmarks. This is engineering specific features. And I think this is fun. This is engineering. It's not leading us closer towards life or towards creation of a cell. I think towards creating a cell, again, we should start simple as possible and let build a system which can self-replicate so that evolution can basically kick in. No reason not to play with the toys you have at hand and build hallmarks, right? Like this is fun, and we certainly learn from that as well.
Yeah, interesting. So what are the some of the applications that you think of DNA nanotechnology-based synthetic cells?
So, I mean, as I said, I think, first of all, what is applicable is DNA nanotechnology. So all of the hardware, the parts that we build, are somehow applicable towards biological systems, right? And we have several examples from our group. One of them being, how can we control cell-cell linkage, for instance, cell adhesion strength, using the same type of linkers that we've used to link, for instance, actin to the cell membrane inside our synthetic compartment, right? So really using exactly what we learned from the minimal. We can see our synthetic cells basically as a minimal model system where we test components for membrane remodeling. We test our ion channels, we test our cytoskeletons, we test structures that remodel membranes, and then we know exactly how they behave. This means we can very well apply them to biological systems as they are. So this is, I would say, the immediate application of the synthetic cells that we build with DNA nanotechnology. Of course, the more long-term application is, okay, if we have a minimal synthetic cell that's based on, whatever components it may be, then we can use directed evolution to evolve that cell towards a given function. And that could be, acting as a synthetic macrophage, or that could be, doing some other useful things, producing a certain chemical or medicine or whatever component it may be. So then we can use directed evolution to make our system do what we want it to do, basically. But for now, I would really say, well, what's applicable about this is, well, just think of the synthetic cell as a model system in which you test the things that you can directly use.
Yeah.
That would be my answer.
Fascinating. And then what do you envision as the future of the field? I mean, so you think that people should continue with DNA RNA based origamis, or there is some space for proteins as well?
There's always space for protein. As I say, first of all, don't be dogmatic about anything, right? There's always space for protein. I mean, I think we should think of the concept of interoperability, right? We should think of, what components can we use to build a minimal functional system? And this has to be as simple as possible, right? And so I think there will certainly be the crowd, and this is very valuable, who is trying to build a synthetic cell according to the central dogma. So encapsulate transcription translation systems and, basically kickstart life like that and end up with a system that's capable of evolution. Then we should at the same time be pursuing different approaches which may not use the native components that a cell uses, but uses more, is even more heavily relying on engineered components, so to say. And there's no reason not to pursue these approaches in parallel. It is possible that the components can in the end talk to each other, so we can unite a little bit the approaches, but it's not a must, right? It's also fair that Life 2.0 cannot talk to Life 3.0, and that's also fair. But since we do use things like DNA, RNA, nanotechnology, I think that, there is a lot of compatibility with proteins. Just think of aptamers, right? Aptamers are DNA, RNA structures which can bind to proteins. So inherently, in some way, one world can communicate with the other. So I would say yes, definitely. If it's the simplest system you can think of, if it's the basic synthetic cell that we all dream of, I don't know. So I don't know if it comes at the beginning or if it comes at the end. Let's see.
Yeah, and what are some of the other fields that really fascinates you? It gives you insights into what you want to pursue in the future.
I mean, as I say, so there are certain fields which have developed technologies which are extremely useful for us. So one field is microfluidics, directed evolution, DNA origami, DNA nanotechnology, RNA origami, RNA nanotechnology. a lot of the work that's being done now in what people would call systems chemistry or also chemical biology is extremely useful for us. Biophysics in terms of the models, simulations, molecular dynamics is like the dream microscope that I always wanted to have, like being able to look at your system in higher resolution. So all of these, and I'm sure I forgot some, it's not an exclusive list of disciplines that can contribute towards our goal of building a synthetic cell. At the same time, I think also, like even going beyond this, I mentioned the field of systems engineering, which I think a lot of the design paradigms have already been developed for, for completely different systems, right? Systems engineers think of the space station or something like that typically, right? But a lot of the principles about engineering complex systems are also concepts that are applicable to us. So looking really beyond our own disciplines is extremely helpful, I find. And looking beyond disciplines also means looking at ethics, looking at philosophy, looking at, society and science communication, because in the end, if we say we want to build a cell, this is going to raise public concerns, and our field is depending on public opinion. We've clearly seen this in the COVID-19 crisis, right? Where, lab origins of COVID-19 were constantly in public debate. And, if we can construct a synthetic virus, or people think so, then why not would a synthetic cell be have as drastic consequences, right? So we have to talk and be in constant exchange with the public about the things that we do, and we have to understand, have an understanding about ethical concerns. We have to start thinking about when certain ethical principles apply, to our synthetic systems that we build, right? And right now it's a bag of molecules that cannot do much, right? But ultimately what we do is gain of function research. So we have to think about these things. And, of course, I've told the anecdote of Ned Seaman. Art can be a great inspiration to scientists, right? So, if you ask about the other disciplines that I find fascinating, it's almost an endless list, and literature, of course, is a great, is really great, it's like sometimes you're inspired by science fiction in a way, right? So, yeah.
Yeah, I mean, especially if you're working with the engineering systems, you need some sort of imagination, right, to come out of these to come out of these standard solutions that you get, like to really bring something interesting, something novel.
It certainly brings in creativity. And I feel like creativity in the field is really, is the driving force. So, just being creative and seeking creative solutions beyond the obvious, this is what's going to lead to the really new and innovative types of science.
Yeah. Okay, and how people can read more about your work, if they want to approach you, what's the best way to do it?
So probably our group website is a place to start. It's got all our publications. So that's www.Göpfrich Group, GOEPFRICH group.de. So I know I have a terrible name, difficult German name. So that's certainly a good starting point. You find my email there. I'd be very happy to hear from you and to continue the discussion.
Yeah, sure. I will put the links in the description of the video. So if people have any concerns, questions, they can reach you. So thank you so much for accepting the invitation. This was a great conversation, and I hope that it'll motivate more people about DNA origami, DNA nanotechnology, and synthetic cells. Thank you so much.
Cool. Thank you so much.
Yeah.
Bye-bye. Bye.